Hello everyone. In the last class, we learned about some of the basic terms used in genetics. In this class, we will learn about Mendel's principle of inheritance. In this module, you will learn about alleles of a homologous chromosome, true breeding lines, and inheritance of one gene. Alleles of a homologous chromosome. Alleles and homologous chromosomes have been discussed in the previous lesson. Link is there in the description box. You can watch it for more clarity. Now let's move on. I have one example for you here. In this image, there is a homologous chromosome of parent 1 and parent 2. Gene locus or area for eye color is marked for you with a brown box. This brown area in the chromosome is going to code for eye color. In humans, the dominant and recessive trait for eye color is brown and blue respectively. That is, brown is dominant and blue is recessive. In the chromosome of parent 1, the brown area is marked as capital B and B, which is dominant for eye color. Similarly, in parent 2, this area has lowercase b and b, that is recessive alleles. Therefore, alleles for gene of eye color in this pair of homologous chromosomes is going to be B and B, one capital and another small. Let me ask you with a few more examples. In this set of chromosome, parent 1 has alleles with both capital A and parent 2 also has both capital A alleles. Remember, capital A is nothing but it is showing dominance of one particular character. So the allele for this pair is going to be capital A. We call it as homozygous dominant. In the next set of chromosomes, parent 1 has both dominant alleles and parent 2 has both recessive alleles. Alleles for this pair are going to be one dominant and one recessive, and you know that we can also call them as heterozygous dominant as it has one dominant allele. All this homozygous and heterozygous has been discussed in the last lesson. In the last pair, the allele combination is going to be recessive in both the cases, as both parents have lowercase a, that is recessive trait. So the allele A and A will be called as homozygous recessive. This means that their trait is not going to be expressed if dominant allele of another parent are present. True breeding lines. These are the plants that have been generated through repeated self-pollination and have become homozygous for a particular trait. Homozygous traits were developed with alleles either as both dominant or both recessive. This trait is then passed on to the future generations if bred with another true breeding plant. Suppose a tall plant with dominant alleles is bred with dwarf plant with recessive alleles. So the traits of tallness or dwarfness will be passed on to the next generation. We will see in a while which is going to express and which is not. Significance of true breeding lines. They have two identical alleles for a gene. So, in a genetic cross, each parent contributes only one form of a gene, making inheritance pattern more detectable and easy. This is a cross between a pea plant with true breeding green pod, that is, the alleles of this plant are going to be, say, G and G as both capital, and another pea plant with yellow pod with alleles as lowercase g. In case of pea plants, green pod color is dominant over yellow pod color. If we want to find out the genotype of offspring, we know we will plot a Punnett square. The offsprings produced are going to be heterozygous dominant, that is, with both dominant and recessive alleles. So, the trait is going to be expressed here is green pod color. This is why true breeding lines are taken, so that the parental character is seen in the offsprings easily. Mendel studied inheritance of one gene in pea plants. Mendel worked on the 7 traits in Pisum sativum. He chose true breeding lines with two contrasting forms. Pea shape that was available in round and wrinkled form, pea color in yellow and green form, flower color as purple and white, flower position as terminal or axial, plant height as tall or short, pod shape as inflated or constricted, pod color as yellow or green. Out of these, we are going to take plant height as one of the character, that is one gene. Mendel studied inheritance of one gene in Pisum sativum. He crossed true breeding tall plant with true breeding dwarf plant. Seeds were collected and grew after this cross to generate new plants. The first generation that was obtained was given the name as filial one generation. He observed that all the progeny plants were tall. It was concluded that F1 always resembled one of the parents. The trait of the other parent was not seen. As you can see in this picture, the F1 generation which is obtained are all tall and no short. Now Mendel self-pollinated the F1 plants. The plants that he started his experiment with, the true breeding tall plants and true breeding dwarf plants. The second generation that was obtained was given the name as filial 2 generation. It was noted that some plants were tall and some were dwarf. He came up with a conclusion that second generation resembled both the parents. The conclusion of the above cross was the tall and dwarf traits were similar to their parents and they did not show any blending or mixing of character. All offsprings were either tall or dwarf, as you can see in this picture. Mendel concluded that something is being passed on from parents to offspring through the gametes. He called these things as factors, genes as we call them now. Genes, it was concluded that they are the unit of inheritance. They contain some information which is required to express a particular trait. For example, height of the plant, that is tall or dwarf. Some important questions are given for you all. Also, there are topic-wise worksheets available. Kindly leave a comment in the comment section if you wish to avail the same. In the next module, we will learn about monohybrid cross. So don't forget to like, subscribe and comment below. Also hit the bell icon so that you will be notified every time I upload a new video. Thank you.